Hello, good morning, class.
Good morning.
So today we are going to be commencing on a new topic on genetics, which is the introduction to genetics. And I'm pretty sure that most of you have a background on a little bit of genetics from your secondary school learning. So the learning objectives of this day, of this lesson, will be as follows. We will start by defining genetics and some other important terminologies as used in genetics. Then we will move on to explanation of four main branches of genetics. And finally, we shall discuss the history of genetics and the Mendelian principles of genetics. So without further ado, let's look at the definition of genetics and some other terms. And before we start, I'd like you to give it a try as to what you understand by the term genetics. Anyone who can try that? Yes, Melly?
Genetics is the study of inheritance and variation.
Exactly, that is very good. So, genetics is basically the study of genes, genetic variation, and heredity in organisms. And when you talk about heredity, we simply mean the distribution or the passing down of the genetic characteristics from the parents to the offspring. So the next term we are going to define is a gene. And a gene is actually a region of DNA, which is also known as the locus, that is located on a chromosome that specifies a trait. And a trait is what we commonly refer to as a characteristic. Then the next term is an allele, and this one is quite a common term. An allele is an alternative form of a gene that can occur at the same locus or place in the genome. And basically, the most common example of this is that you can find that probably somebody has a gene for tallness, but at the same time, that person carries a gene for shortness. And that is, those two genes are alternative forms of one characteristic, which is the height. Then we look at the other term, which is locus. And the locus refers to the location on the genome of a gene. We'll move on to the other term, which is haploid. And this one is basically the condition of having only one set of chromosomes, while diploid refers to the condition of having two sets of chromosomes per cell. Yeah. So we move on to the next objective, which was the branches of genetics. And the four main branches of genetics are as follows. We have classical genetics. Which is basically the branch of genetics that is based on the visible results of reproductive acts. So if you're told to give visible characteristics, what can you think of? Anyone who can think of a visible characteristic? Anyone?
These are external features which can be seen in an organism.
Yeah, exactly. So an example is the height of the organism. The other example is the body weight, or many others. I'm sure you can think of a few. Then the next branch of genetics is molecular genetics, and this one basically encompasses around the order and trade of genes, and it is very common in molecular biology and genetics. Then we move on to quantitative genetics, which basically is a branch of population genetics that deals with continuously varying phenotypes. So when we talk about phenotypes, these are characteristics that can be seen with the eye. So from there, we quickly move on to the history of genetics. As you all know, everything has a beginning. So when it comes to the history of genetics, at this point, I'm going to be talking about various timelines. And the first timeline is in 1859. In 1859, where you find Charles Darwin. Charles Darwin proposed a famous theory of evolution. I'm sure you've heard of this in your secondary courseware. And you find that he came up with the slogan that there is survival for the fittest. And you find that when he talks about survival for the fittest, he simply means that those organisms that are not best suited to the environment are going to be phased out, and the ones that are best suited are going to live long enough to pass their genetic characteristics to the next generation or to their offspring. From there, we move on to the other timeline. Which is in 1865. And in 1865, a man known as Gregor Mendel, while experimenting on pea plants, discovered that traits can be passed from one generation to the next. And in this point, he formed some predictions that were coupled with statistics. They had statistical evidences. And he tested and came up with dihybrid and trihybrid crosses in pea plants. So when we talk about dihybrid, we simply mean 2 crosses, while the trihybrid means 3 crosses. So we move on to the laws of dominance as brought forth by Gregor Mendel. And I'm going to ask one of you to go through that slide and read whatever they can see. Anyone who is willing to do so? Anyone? Yes, Onesimus?
The law of dominance. Some characteristics tend to be dominant and be expressed, while some are recessive and be suppressed. And the second law of segregation describes how parental factors separate in the gametes or the sex cells. And then there's the law of independent assortment. Describes how hereditary factors assort independently during the production of gametes. Gregor Mendel was known as the father of genetics.
Exactly. So thank you very much, Onesimus. So as you can see, those were the 3 laws that Gregor Mendel came up with. And the first one was on dominance, which is you find that the gene that is actually dominant is the one that is expressed. Just like I was saying a few minutes ago, you find that there's a person who would have genes for both tallness and shortness. So if this gene for tallness is actually dominant, then that is the gene that will be expressed. And so that individual will be tall. But if the other way around is true, if you find that the gene for shortness is the one that is expressed, then that person will be short. So the other law that he came up with was the law of segregation, which basically is based on parental factors separating from the sex cells. Then the last one was the law of independent assortment, where you find that heredity factors were assorted independently during the production of gametes. So the last thing we're going to look at in this lesson is a chart on the Mendel's experiment, and that is the chart. So Mendel was able to experiment with pea plants, and that is what he was able to come up with. So as you can see on the chart, there's the dominant trait and the recessive trait. So for color, we have purple as the dominant and white as the recessive. Then for plant height, we have tall and short for the recessive one. Seed color is yellow and green for the recessive. while the seed shape is round and wrinkled for the recessive. The pod color is green in the dominant trait and yellow in the recessive trait, while the pod shape is inflated in the dominant trait and flat on the recessive trait. And the last thing is the flower position. You find that in the dominant trait it is axial, and in the recessive trait it is terminal. Yeah, so you find that basically when it comes to the ratio of representation between the dominant and the recessive trait, it is usually a ratio of 3:1. Yeah, so from there, any questions, any clarifications, any complaints? Yes, any question? So if there are none, then I beg to leave. Thank you so much for attending the class. So we will meet in our next session. So have a nice day.
Thank
You
you.
too.
Thank you.